Okay, I guess we can get started now. I saw there's a comment in the chat that there's no sound. Now there's going to be sound, hopefully, but let me know if there's still no sound. So thank you again for coming, everyone. So last time we talked a little bit about the background of RNA sequencing, sort of different approaches, different considerations. And so this time we'll do a little bit more hands-on, sort of trying to get some data going from some files to hopefully some results. But again, as last time, sort of let me know if anything is confusing or unclear, and we can we can stop and go over in more detail. So just a little bit about some of the tools we'll be using. Unfortunately, this is the protocol I'll show you is streamlined for Big Purple. For prints, it's a little bit different. Since I don't normally use prints, I haven't had a chance to really set it up for prints. I'll try to discuss sort of how you would adapt it to prints, but sort of the default workflow is for Big Purple, and there's some Big Purple specific considerations. So those of you who are in prints, I do apologize, but I'll try to cover both bases, both sides, maybe at least to some degree. Obviously, you should have a SSH client, such as the terminal on Mac or PuTTY on Windows. And what I like to use is a graphical FTP/SSH client. So something like CyberDuck or Fetch or FileZilla, because yes, the terminal is great, but I think it's a lot easier when you can actually see as the files are being generated and what they are. And so, for example, if you have images, no matter how good you are with a terminal, you will not be able to see them with the terminal. So that's unfortunate. And I'll show you kind of that part as well. So basically, what's our general goal with RNA-Seq? Usually you have some raw data, which is probably represented as FASTQs, and then you want to get some results. Or really, ideally, you don't really maybe not care about the results, but you really want figures so you can publish and flourish. Unfortunately, there is a little bit of a gap between these two parts. And normally normally people expect it to be kind of a straight line. You push a button in an ideal situation and it gets you from one to the other. In reality, sometimes that straight line is not so straight. Sometimes there are a lot of roadblocks and there are a lot of things that you don't expect. As I've shown last time with one of my slides, so during the RNA-Seq analysis roadmap, there are a lot of steps, a lot of things to consider. And at each of those steps, each of those parts, you can get tripped up on something and you can cause different problems. So these are just some of the classic RNA-Seq, kind of a classic RNA-Seq workflow. There are a few different options you have. And so the option that we'll take today is sort of this leftmost column, where we'll do star to align, feature counts to count, and then DSeq2 for the differential expression, which then Tosh will actually address in more detail next week. But one caveat that I do want to mention is that traditionally, people who don't do analysis assume that most of the analysis is actually analysis, but it's not. So most of it is, this applies to data science, not necessarily bioinformatics, but we can use them maybe interchangeably sometimes. Most analysis is not analysis. So most of the time you spend is actually cleaning and organizing the data or collecting the data. Maybe for sequencing, in terms of sequencing, maybe you're not collecting the data that much as an analyst, but there's still a lot of cleaning and organizing data, and also quality control. Sort of when you get the data just because you have a FASTQ, it doesn't mean it's actually any good. And so I would say the most important part is not just the basic workflow, but also making sure that all the steps you're confident in and you're confident in the actual data. And that's a little bit harder to summarize in a very, to really cover in a very basic workflow. And then another step is pipelines. So what is a pipeline? That word is actually frequently abused in our field, or in biology in general. There are a lot, I mean, what defines a pipeline? It's a kind of, some people just call a simple script a pipeline. And then also there are a number of different very complex workflows and toolkits that are available. So here, for example, there's sort of one resource where they keeps track of some of these different pipeline options, and this is just taking the ABCs, and there are already a number of different versions available, and sort of like, so there's sort of certainly no shortage of them. And of course, there's been at least one review of different bioinformatic pipeline frameworks that looked at sort of some of the more common ones that you may or may not have heard about before. Certainly sort of the people on the bioinformatic side of things may have heard of a lot of them. And really kind of, I think sort of the problem here is that sort of the first column is the ease of development. So really a lot of these pipelines are really targeted for development. So if you're a developer, that's one thing, but sometimes if you're a biologist, maybe that's not your top concern. So this is where I come in. Basically, for those who are not aware, Sleep and Slide is a classic game. That may be some cultural issues there, if you're not aware. And so this is the data set that we'll be using today as an example. So this is a mouse data set where they had wild type and tattoo mutant animals, and then they compare the macrophages in the two. So this is it doesn't really matter particularly what exactly they do for this purpose, but really it's just a very clean experiment where you have two very distinct groups, and it's a relatively decent quality data set. And so basically we'll just jump to the demo right now. So I set up a web-based page where you can follow along, so you don't have to, since you obviously can't copy and paste from the Zoom screen. So you can so if you go there, you'll have the same instructions and just slightly more text. Oops, where are we? So if you go there, this is the page that you should be seeing. This is the demo for Sequence Slide. So Sequence Slide is something that I slowly developed over many years to basically streamline analysis over its sort of various protocols, including RNA-Seq, but there are some other routes that are available here as well. So what are we going to do? So this is, as I mentioned, this is the wild type versus tattoo knockout mice. I actually already on Big Purple, I already downloaded these files, and there's a directory with them. But first, before we begin, some first steps to basically prepare the pipeline. You first should create a project directory. Well, I'll make it. So I will zoom in here. Hopefully this is good enough to see for everybody. So we'll make a directory called class run. We'll cd to that directory. So right now, there is nothing in that directory. So one of the tools that you kind of have to use for this is Git. You don't actually have to know much about it. We're using it just to kind of download the code, and it doesn't actually do anything. So we take module add Git. We can just paste it in here. And so now Git should be in your environment. And then the next step is to download the code, which is using Git Clone. Again, you don't really have to know Git very much. Just this is basically just downloading the code, and you can see that it's downloaded. And now when you do ll, which you can see that there's now the SNS directory that's available. Alternatively, I like to use fetch to visualize what data you're looking at. And so here, for example, we created the class run directory. And now when you refresh, there is the SNS directory there, but otherwise it's empty. So the next thing is to process the individual RNA-Seq samples. So first we have to specify which genome we're using, because no matter how smart any kind of tool or pipeline is, it does not know, it's not going to know what species you're sequencing or what reference to use. And so we'll here generate settings. Oops, where are we? There we are. And then and if everything works, I was able to find the reference directory. And so as I mentioned last presentation, yes, there are kind of the tools that you're using, but besides the tools, another consideration are your reference files. And although there are many tools that are well described, once you start to try to figure out which reference files you want to use, you run into all kinds of different issues. And then depending on the genome version, depending on the reference source, there could be a lot of different caveats. So it's sort of trying to streamline downloading and generating the references is sort of a big task in itself. So which is why I keep on Big Purple some of them in my own directory that will be used here. But and so, as I mentioned before, it just shows you that something worked. If you go back to fetch, you'll see that now we generated the settings.txt file. And what's really nice, if you're, for example, on a Mac, if you're using something like Fetch or CyberDuck, maybe some of the other tools, if you click on the file and you press spacebar, it'll actually give you a preview, which on a text file is not particularly helpful, but on an image file, it's going to become a lot more interesting. So here you can see that it filled the genome directory and the reference FASTA file. Then the next step would be to find your FASTQs in some directory and then create a sample sheet out of them. You can do it manually, or there's an automated version, which you press, and then it looked in this directory and found 8 different files here. And now when you do ll, you can see in addition to settings, now there's a samples.fastqraw CSV. What's really great, and so again, I'll highlight how great fetch is again on the Mac, if you press spacebar on a CSV file, you can very nicely see a table where you can see what it made is a 3-column table with the samples in the first column and then the FASTQs in the second column. And if these were paired end FASTQs, so read 1 and read 2, you would put read 1 in the first column and then read 2 in the second column, and then read 2 in the third column. And then it's also try to automatically extract the name. In this case, it worked well. Sometimes it doesn't. Of course, you can always go into the samples FASTQ and edit it and sort of fix the names, add or remove samples. But essentially, this is going to be our experiment. These 8 samples with the FASTQs that come from these locations. And then the third step, so first you generate the, figure out, to specify the reference, then you specify the FASTQs, and then finally you just run the pipeline. In this case, we're using the RNA star, what I call route or workflow, but there are different ones available, which you can read about later. So when you run, what will happen is that it shows you kind of some basic information. and then it starts submitting these samples. And then because Big Purple is relatively busy lately, I don't know how many of them will actually go through for all of us, but hopefully at least for some of us, some of them will go through and we'll start seeing the results right away. And so now if I go back to fetch, let's see if anything is actually happening. Nothing is happening yet, which is not surprising, but given the stress load. And so the first few are submitted relatively slowly. And then after the first few, it'll go much quicker. First few are just sometimes when you first submit, you'll start seeing weird things. For example, if your sample names have weird names, you might want to cancel really quickly because you don't want to run the rest of them. And so now when you do, yes. So someone asks, what is RNA star doing? That is an excellent question, and we'll get to that in a second. So right now we can see that now things are happening. Now, for example, we get this FastQ clean directory. But yes, as the question said, and what exactly is RNA star doing? So we'll go back to our presentation for a second. You'll have a record of it. So to keep track of progress, there's going to be logs sbatch directory, which are basically the sbatch or sort of your standard error, standard output files. And then there are various summary files. And the most important one is probably the summary combined, which is going to be the combined summary files across the different samples and different segments in one. And specifically, which steps are included. And so these are the steps that are included and what RNA star is doing on a very broad level. So we'll trim the adapters and low quality basis. So basically we'll take our FastQs and we'll try to, in a kind of somewhat unbiased way, to get the reads that are high quality reads, which means sort of without adapters and where the bases are good quality. We'll also align to the reference genome with star, as I mentioned. You can also check against common contaminants, normalize the genome browser tracks or bigwigs, check the distribution of the bases within the transcript. So to see if there's a 5' or 3' bias, we'll do it with Picard. Determine if the library is stranded and the strand orientation, because that becomes important later on. And then finally, generate gene samples counts matrix with feature counts. So really, the two steps that are kind of most crucial is the alignment and then the quantification. But of course, the other steps that are included are also important. And if you have really great quality sequencing, you can definitely get away with just these two steps. But sometimes you don't know what kind of quality you have. You don't know what's in your samples. So some of these other steps really help you figure out what is in your samples and improve their quality. So, but that's really broad. So what exactly happens? So when you go into the SNS directory, this gives you all the code that is actually being run. So here it's split into two main directories. So there's the routes directory, and within routes there are separate scripts for different routes or workflows. So for example, there's one that's RNA star. Then you can open it here, and then essentially take some inputs, which is sort of your project directory, which sample and which code. And so each sample is going to get submitted as a separate RNA star process. And then these are just the various settings. But really, so what kind of for each sample, we'll have kind of a few different steps or segments. So for example, the first step here is FastQ clean, which submits the sample to the FastQ clean segment, which basically checks that the FastQs are, for example, if they're the same size. Sometimes you have paired end reads. You might have, let's say, 10 reads in read 1, but 11 reads in read 2. Clearly something went wrong. for example, if you were copying files, there might have been some error on the way. And so that step sort of does that, and also gives you a summary. The next step is FastQC. Then we'll do FastQ screen, then trimming with Trimmatic, alignment with star, generating the bigwigs, doing Picard RNA-Seq metrics, and then I'll determine the strand of the reads, and then quantify the reads with feature counts. And so all of these steps are actually, all of these segments are actually have their own file in the segments directory. So really what you can do theoretically is if you go to segments, and we'll take something like the align star step. So this is a dedicated bash file that's specifically for running the alignment segment. So here it just takes the different arguments that you give it. But really, if you were working on prints, for example, or a different server, or you kind of wanted, or you just wanted to do this more manually, you could just take these commands here as the example. So for example, some of these, so for example, it checks whether sort of the input files are actually existing, whether the output already exists. And then if you keep scrolling down, eventually you get to the actual star command. And so here, for example, here's the star command. And so is this maybe excessive? Perhaps. And so if any of you have used star in the past, for example, the default command that you can use is actually, you don't need most of these parameters. So usually you just need the two FastQ files and the output, and sort of the output base name, and your reference genome. But what you end up learning over many years of trial and errors, that sometimes there are additional tools that you want to use later that expect a certain type of BAM file. And so kind of over time you start to add all these various other parameters just to make sure that the BAM file that you get is actually compatible with all these other various tools that you might want to use later. And this part is not, although technically it's documented somewhere, it's sort of trying to figure out kind of all the different tools, how they work with each other. It is a little bit of a trial and error over a long period of time. So for example, if you want to call variants from your RNA-Seq later, you may want to use something like GATK toolkit, which is common in the whole exome, whole genome world. But it does expect the files to be formatted as certain, the BAM files to have certain certain information in them, which you kind of have to set here. And of course, there are a few different tools. And so basically, you can run this star command manually. You would just have to replace the variables with the with the relevant information. But you know, this might be kind of, and then it does some post-processing, which is try to summarize the output. So for example, star generates counts, and then here it tries to summarize that counts in a nice table. Or it also tries to determine the strand. So as I mentioned in the last class, you can have stranded libraries or unstranded libraries. And even if it's stranded, sometimes you don't know if it's forward strand or reverse strand. Additionally, even if you do know, depending on the manufacturer, depending on the tool, what some people call forward and reverse strand is also different because if you're, some people refer to it in the context of mRNA, and some people refer to it in the context of cDNA. And so so you can have two opposite kind of answers to this question, even though they're both correct, that doesn't really help you. And so I sort of and then kind of trying to estimate it based on the actual output is the safest way to know that what you're looking at, sort of the strand that you're looking at, is really the strand that was being sequenced or being targeted for sequencing. And then it also generates the summary file. So I'll close that. And I'll go back to this main directory where you can see things are happening and more files are being added. So for example, there's the first summary for example, there's the summary file that gets generated for every, or for many of the steps. And so now if you can do spacebar preview, for example, again, it gives you kind of a nice summary. Hopefully it's not too small over the zoom window. And then here it basically tells us what our samples are, and then how many reads were read 1 and how many reads were read 2. Usually it's not, this summary is not particularly exciting, but you basically see that all of your samples had read 1 and none of them had read 2, which is expected, but it's nice to see. You also see how many reads we had. So I actually downsampled this data set to only have 10 million reads for most of the samples. So and then you can see here that they are indeed 10 million reads. This is just to kind of speed up the analysis. For some of the samples, it's actually less reads. Again, just to have some variety. But normally you kind of do want to have, for all your samples, to have a similar number of reads. Or even if you, regardless of your expectation, you do want to have here the number of reads that you are expecting. So if you were, if you paid for 10 million reads and you're not getting 10 million reads here, this is sort of a concern. But what else is it doing? Again, sort of going through all the bash files, if you're kind of not familiar with that, which is probably most of you, that might be a little bit overwhelming. So what you can do is, in your directory, there's the logs sbatch directory where all the files are stored. So we can do cat logs sbatch 20. So here we have 8 files that are running, and then we have 8 output files. So for each sample, the entire RNA star route is going to generate output. And so this is basically a giant, well, it's not so giant yet, but it's basically a file that gives you all the output. And so here just tells you that we ran this route. Then for each segment is going to have its own summary output. And then the command that was run. So here, for example, the command that was run is we just basically use the symlink to link to this initial directory to put into the fastq-clean directory a link to the initial file. And the reason why it's a link is because why duplicate a file if you don't need to, because these fastq files are big, so we should be considered to your storage. Of course, if you had multiple files for each of your samples, so for example, if you ran it on multiple lanes, then you would want to concatenate them into one, and so then this step would have done that. But if you start with one file, there's no reason to duplicate it. And then again, it just gives you a little summary, what file you ended up with, and then how many reads you had that you saw in the summary file. Then there's the QC fast QC segment, which is for running fast QC. It gives you a little bit of information about sort of which fast QC binary you're using and which version of the tool that is, and then what the different inputs and the outputs were. So for example, this FastQ file is the same one we had here, and this is the same one we're feeding into the FastQC. And really, as I said before, if you look in the bash file, it can get confusing because there's a lot of sort of helper code to kind of make sure everything is fine and to catch various errors. But really what it's doing, if you want to summarize it, is really just running this one command. And I think maybe you already see the pattern, which is normally when you first start doing these kind of, any kind of bash programming or any kind of analysis, you start with pretty simple commands, such as, fastqc, and then, you specify your output directory, and then you specify your input fastq, and then you run it. And over time, you might want to automate it. And so that's where you end up with something like a bash file. And I think you guys have done this in some of the earlier classes, which is really kind of writing a bash file where you can have different parameters, and then you can run the tool in a more automated way. And so then you might want to check if your inputs and your outputs are valid. And then maybe you want to do multiple samples at the same time. And so basically that's how you end up with SNS at the end, just over time, slowly adding more and more automation features. But at the end, you really are just running a series of commands, run one after another. So then there's another command for another step for fastq screen. And oh yeah, and I guess I should mention that, yeah, you could just sort of run all of these manually one by one. It's going to take some time, but sort of why not automate the process? And there's another argument to be made about whether sort of, is this maybe too easy? So for example, from my standpoint, should we really be doing this one by one, or should we kind of do this automated protocol, which I don't have a problem with. I think sort of everything, all various tools are built on top of other tools. So really, there's kind of no reason to sort of draw arbitrary distinctions about sort of what is kind of the low level enough protocol. So then the next step is fastq screen. And again, it's just the command is, that's the command. It's fastq screen. Then you specify sort of which aligner you want to use, the threads. We're actually using a subset of reads because it's just a QC, it's just a quality control step. And then using check which configuration file we use, what the output directory is, what the fastq file is. And so what is fastq screen? This is where this preview becomes nice. So it also generates a summary. So for example, here's the fastq summary. So here for each file, for each sample, it'll generate a separate plot. They're shown here together. Since we're in Zoom, it's a little bit probably hard to see for everybody. But where is it? But if you go to this QC fastq screen directory, which you probably normally don't need to, but since we kind of just want to show it really big, it'll show up here. Or we can actually open it with preview, so it's a little bit even bigger. So what it does is it takes that fastq and it aligns it across multiple genomes and transcriptomes. So for example, here we can see that almost all of the reads here are aligning to the mouse genome, which is great because these are mouse samples. So you would expect most of them to come from the mouse. Sometimes you don't get close to 100%. Sometimes you get much. Trying to move the chat here. Sometimes you get much lower. So for example, if you're the most simple and the most reasonable examples, if you're working with xenografts, you might be inserting a human tumor into a mouse, and then when you're sequencing, hopefully you're getting mostly human reads, but sometimes it's you get some mouse reads. Sometimes you get mostly mouse reads, and then you know that maybe you're not properly collecting your sample. Sometimes it's more, sometimes you get more weird contamination. One nice thing that I do here, for example, is checking for Mycoplasma contamination. A lot of times you do get Mycoplasma contamination, and a lot of times various, the biologists who have the samples may not even be aware that they have Mycoplasma, and sort of doing this in silico screen, you can actually not only troubleshoot this one experiment, but also potentially save many future experiments. And a lot of time, and there have been many occasions where I found Mycoplasma, and then people went back and checked sort of in a kind of more experimental approach, and they did find they did actually find Mycoplasma in their tissue culture. So so that was kind of a good result. I mean, or at least it's a positive result. It's something that can be resolved. You can also have adapter contamination. So for example, if there's a problem with your input, you might have a lot of adapter dimers instead of actual DNA, cDNA fragments, and so that's also a problem. And then finally, there's no hits. So if none of the reads align to any of these categories, you'll end up with a read that's not any of these. So for example, if you use the wrong genome, for example, or if you made some mistake, or maybe the sequencing facility made a mistake, or your collaborator made the mistake, and then there's just sort of other various species. So, and also within the mouse here, we have mouse genome, so the full genome. There are also two other mouse classifications here for RNA and ribosomal RNA. So ribosomal RNA is something that you do not want to see a lot of. If you're doing poly-A selection, you're probably not going to have a lot of ribosomal RNA because just due to the way the library is constructed. If you're doing a total RNA library, what may happen is that you can get sometimes the ribodepletion doesn't work very well, and then you get a lot of ribosomal RNA. Sometimes it's 10%, sometimes it's 20%, sometimes it's more than half. And so if sort of 90% of your sample is ribosomal RNA, which could happen, then you might not get a lot of counts for the actual relevant genes. And sort of this would explain it. And that's sort of a kind of a problem that you can have that's a little bit difficult to diagnose in other ways. And then there's also the RNA subset, which is basically the transcriptome. So most reads are aligning to the transcriptome. There are a few reads that are aligned to the genome, but not to the transcriptome, because although you would like to think that annotation is perfect, there are actually some genes that may be unannotated. And so that could be coming from there. You could have maybe a little bit of DNA contamination. There are various kind of possibilities there, but sort of the possibility that most people get excited about is so we have all these sort of unannotated non-coding RNAs that maybe are interesting. So, but of course that could be very difficult to really pin down, and that requires a lot of additional analysis. And then just the colors here, the blues means it's only aligning to one genome, and the reds mean that it aligns to multiple genomes here. So not surprisingly, everything in transcriptome is red, because everything that's in the transcriptome should be in the genome as well. And what's interesting here is that, for example, here we have a little peak for the Homo sapiens. And so we have about 10–15% aligned to human. But what's really great is that if you look, actually all these reads are multi-mapping. So it's probably the reads that are actually homologous between your mouse and your human. And considering that there may be 10% of them, I think that's fairly reasonable that you would have 10% of your sequence to be similar. Of course, especially because these are short reads. You don't have that much room for to really be very accurate. So the step that we missed before is actually FastQ screen. So FastQC. So FastQC also does an output, and that's probably not as exciting, but again, it's nice to sort of be able to preview it. So you can see here that actually I was wrong. The reads are not 50 base pairs; there are 100 base pairs, and you can see the quality. And I think you looked at FastQC earlier, but this is just sort of just to show you that for each FastQ you can see kind of the quality. It looks like the per tile quality is actually a little bit off. Some tiles, this is tiles on the sequencing flow cell. Some of them are a little bit lower in quality, so maybe whoever sequenced it should look into their instrument to see if maybe there was a little bit of fluidics issue there, although it's not particularly bad. I mean, I've seen much worse, but there's definitely some tiles that are worse than others. And then some of these other metrics are a little bit more difficult to necessarily interpret, but they do look fine. And then you have, not surprisingly, you have some adapters. So for example, as you go across the read length towards the end of the read, you start seeing some adapters where if your cDNA fragment was too short, then the sequencing continued into the adapter on the other end. So that's fine, which is why we do trimming to remove those ends that are actually adapter ends. And so when we first looked at this file, it was still running FastQ screen, but let's look at it again. So we're still on FastQ screen, and then the next step was using FastQ trimming, doing FastQ trimming with Trimmomatic. And as the other steps, you can actually see the exact command that's being used. So you could theoretically take this command and run it manually if you don't want to run this automated pipeline, and really it's just sort of running the Trimmomatic Java jar file, and then you specify the various parameters, and then hopefully you get an output. Some of the samples are already finished, and then there's a nice summary table that gets generated. It's only 3 samples for now, but you can see that it shows you the number of input reads, which is, so not surprisingly, this really small sample finished really quickly, but then also 2 of the bigger samples finished where they have 10 million reads. And then after trimming, we actually threw away about 2-3% of the reads, which are low quality. So having gotten rid of them, we can be more confident that the reads do remain actually good quality reads, so the data that they're giving you is actually reliable and reasonable. Let's see what else. So actually, while we were talking here, the FastQ trim actually finished, and then it went on to the star alignment step. And again, it gives you some information about the tools that are being used, which star is being used, which SAM tools is being used, which genomes are being used, and then the actual command where, again, you could just take this command, paste it in, and it would give you the same output. Or what I forgot to mention is, of course, these are all bash files. So if you go into any segment and, for example, you want to change some of these parameters, again, all of the files are just simple bash files. So you could just modify the command in there and then rerun the project again. And then it is going. I think we're still a little bit slow. I actually ran this earlier today just because I thought that maybe it would take too long. So if I actually go to a separate direction, so this I just did about an hour ago, so it's very simple, very similar. So then another step that we make is this RNA-Seq metrics from Picard, where for each sample, it will take the most highly expressed transcripts, and it will look at the relative coverage along the transcript from the beginning to the end, from 5 to 3 prime, and then it's essentially normalizing each transcript from the beginning to end, from 0 to 100. And so normally in a perfect RNA, in a perfect world, your transcripts would just have this sort of a sad face, where obviously you're not going to capture a lot of reads at the very end of the transcript, but sort of in the middle, you should capture, they should be relatively flat. So you kind of are getting the full transcript across all the different transcripts. What can happen is that you can have some degradation, for example, and then if your RNA is degraded, you will start noticing it here. And then, for example, if you're doing poly A selection and you have shorter RNAs, then naturally most of your reads are going to be biased towards the poly A tail. And so you would see it here. I mean, here there's a little bit of a shift, but not not as bad as it could be. So for sometimes, for example, it basically looks like a triangle, or all of the reads are piled on at the end. And then finally, oh yeah, and then I guess I mentioned there's a summary for the star alignment, where you have the number of input reads, the number that are uniquely mapped and multi-mapped, and then them expressed as a fraction. So here, for example, most of the reads, more than 90%, are uniquely mapped, which is great. Sometimes that number can be a lot lower. It could be 50%, which is still sort of within reasonable, but it really depends on on a lot of factors. But obviously, you do want it to be as high as possible. And then, as I also mentioned, all of the summary files are eventually merged into the summary combined, which is a single file that sort of contains all their information. So every time I do RNA-Seq analysis, this is the file that I check to make sure everything looks okay. You can even, it's a CSV file, so it should be openable in Excel. So you can, it's a little bit easier to read for human eyes. So again, we saw some of the previous information, such as the number of reads, sort of the trimming statistics, the number of uniquely mapped. Actually, I'll make this bigger. So the number of uniquely mapped, assigned counts. So here you have two different versions of the, so how many reads were actually assigned to a particular gene by feature counts. So you can see that there is an unstranded option and then a reverse strand. Since most of the reads were on the reverse strand, that is the strand that was done, but sometimes you want to see what would happen on an unstranded protocol. And you can actually see that for some of the samples, the number is actually a little bit higher on the reverse strand because some of the reads where there was questionable whether how it should be assigned. Once you have the strand information, it could be accurately assigned to a specific gene. So the number actually does, in this case, go up a little bit. Sometimes it goes down. Also, the number of aligned bases, which is just reads multiplied by the read length. You can also, so this is what I mentioned before, how do you know what strand it is? So you can actually check with Picard RNA-Seq metrics. You can, in addition to the plots I showed earlier, it gives you the reads, the fraction of reads that are assigned to either read 1 or read 2. And that's not a problem with strandness. Not only can it be forward or reverse, sometimes they call it read 1 and read 2, and there are a few different names depending on the tool. But again, you can see that 99% of the reads are on this strand and less than 1% are on a different strand. So clearly this is a stranded protocol. You can also see how many genes are coding. So here, sorry, how many reads end up within the coding region. So here, for example, we have about 45% within the coding, which is good. And then about a third in the UTR. And then there's some about 20% that are intronic, and then even fewer that are intergenic, which is great. Sometimes if you have DNA contamination, for example, this number is going to be much higher than 2%. And again, sort of intergenic doesn't mean it's actually intergenic. It just means it's intergenic relative to annotated genes. It is possible that all of these are really exciting unannotated link RNAs, although not very likely, but it is possible. And then more broadly, the number of the fraction of reads or bases that are within mRNA, which is about 80%, which is great. And then this is the 5' to 3' bias, which is a little bit easier to see visualized because there you can actually see kind of as it goes along the full transcript, what it's actually doing. So that, and then it also generates the bigwigs in this bigwig directory where you can, and then those are the tracks that you usually want to see at the end. You can put them into something like IGV or UCSC genome browser, and then kind of view how much coverage you have across the different exons and across different genes, sort of, and then down from here. And then finally, I should have mentioned this earlier. If you use SQ, which you covered in previous classes, you can see what's happening. So this is just one way to see. Where you can see is sort of that there are a few samples that are still running RNA star. It's cut off here, but hopefully they are. But everything is running, which is great. And then probably the most important step is as it's running or after it's running, you can check for errors, which is a grep command which goes through all the logs as batch files and checks for error. And normally you will not have any errors, but if you do, then you should check that file and see sort of where the error is and kind of what might explain it, which can be difficult. And then finally, there's a samples.groups file. I'll go back to the actual, we have it in, that gets generated. So this is the project that's actively running right now. So here it's this table where you have all the different samples, and then there's a column that gets added that's called group, where you can fill in the different groups that different samples belong to. So by default, of course, the pipeline wouldn't know which samples are which groups, and maybe you don't know either. For all we know, the samples are actually mislabeled. So it's not always obvious how they're going to be organized. But then I'll go back to the previous run where I already filled it in. And so here, I'll open an Excel to zoom it in. And then I basically labeled each sample by their group. So for example, the wild type samples here, and then the knockout samples. In this case, it's relatively simple, just two groups, very easy. And then once the regular RNA star step that's doing it per sample analysis is finished, you can perform the differential expression analysis. So there's a separate step, RNA star groups DG, to do the differential gene expression. Of course, I can't run it on other samples yet, but I did run it earlier for this particular, for the earlier run, and then it generates this differential gene expression step. So once you go there, there's a lot of different output. And so unlike kind of the primary analysis where really all you need is just to go from FASTQs to a table of counts, here there's a lot more things that you can do, and which is why next week is a whole week devoted on sort of what you can do with DE-seq and sort of how you can manipulate the sort of the inputs and the outputs and the different steps. Here it's kind of the default output where it gives you the normalized counts. So you start with the raw counts. You can open an Excel. So here you have the different genes and the different samples. And so you can see, it's a little bit hard to see on the top because the genes look weird, but if you scroll down, there are a lot of GM genes, which are also not fun. You can see, for example, for all these different genes, some samples have some counts, some have more counts, but these are the raw data. And then these are, and then from that you get various normalizations. So this is just a standard DE-seq normalization. Which is probably all you need. And I think you actually had this table in an earlier class, where again, you have sort of different samples, and now the counts are normalized. So based on the library size. So now sort of these are comparable. So if you wanted to make some bar plots or box plots or any kind of relative expression that you want to compare different samples, you would be able to take these values and do that. And then there's a few different other options like FPKM or TPM, which you might want to do. It also generates PCA. So that's another, so sort of the primary QC once you have for differential expression, sort of to see kind of how your different samples are similar or different from each other. So this is just looking at kind of overall transcriptome differences and trying to essentially summarize the two dimensions that explain most of the variance. And so here, this actually worked out fairly well, where your knockout group is separate from your control group, from the wild type group. If you're working with human samples, it's probably going to be very difficult to have two different groups separate, unless you're looking at something like tumor versus non-tumor. Sort of, if you're just looking at different treatments, it's probably not going to be so sort of the natural biological variation is going to be much higher than your stress. But in here, we do have a nice separation, although one might say that this PEN8 sample and then the PC1, which explains most of the variance, it does seem to be substantially different than any of the other samples. So maybe, there's some problem with it, or, maybe it's natural. It's hard to say. But, if it does look, if you do sort of, if you have additional information that might tell you that the sample should look weird, then this just confirms it. But again, it's not terribly different, and it still kind of groups together with the rest of the wild types. It'll also generate the differential gene expression table. Which will give you for each gene, sort of the log fold change, the p-value, and then the FDR adjusted p-value. So basically the genes that are most changing. Here, you can also do Volcano Plots, which basically just plots the genes on a volcano. You can sort of see the top genes here. It'll also generate some heat maps. So based on various cutoffs. So for example, what are the top 50 genes and how they separate the samples? And here it's pretty good. The top 50 genes do separate, the most significant genes do separate the two groups as would be expected. But sometimes it doesn't happen. So then that's something you should look into and see, and sort of see why that does or does not happen. And of course, there's gene set enrichment, which kind of shows you if there's enrichment in certain pathways for your differentially expressed genes. So for example, here we see that the interferon signaling is increased in the knockout samples. But there are a few different ways to do the pathways. And of course, you'll go through DSeq in more depth next week. But if you wanted to sort of see what exactly that step is doing, in the script directory, there is the differential gene expression step for DSeq 2. It's a sort of it's an R script, and so here you can open it, and sort of you can try to do this kind of one line at a time, and then most of it is just loading different packages, but then it's sort of some of the messages that get output to give you the progress status, but then eventually there is the actual analysis, such as it's a little bit hard to see, sort of like convert the matrix to a DSeq object, and then do the actual DSeq analysis, and then of course this is sort of all the follow-up that happens, but all of this is kind of, you can do one step at a time if you so desire. And of course, if you go to the logs sbatch, the last file is the one that was used for differential gene expression. It sort of tells you where the directory that you have, where it is, what the different inputs were, the counts table and the groups table, which is really what the R environment was. And then kind of as your script was running, here's the output it mentioned. So it tells you kind of the number of genes that were present, what the different samples were, In our case, it's a pretty simple experiment, so all of this makes sense, but sometimes you can have a lot of samples and you might have some weirdness going on there. It also tells you kind of from your GTF what the kind of number of genes that were there and sort of size, and then kind of all the different steps, some basic outputs. So for example, how many genes were with a p-adjusted value less than 0.01, which is 150, which is relatively good. And sort of that's the, yeah, and then this is sort of some of the more standard, more error-like output. But yes, so that is that, and that brings us to 1 hour. So hopefully that was helpful. And that is it for now, but I'm happy to take any questions or anything. Thank you.